disease resistance. From this also we followed the same approach, but then here our approach was not candidate gene based approach. It was based on genome wide marker approach. So we genotyped for all the genomic regions through GBS, and we evaluated for the phenotypes MSB, and we did what you call association analysis. Do you know what softwares can do association analysis? Very good. TASR is one of the very powerful software which can implement this mixed linear model, right, which is required for association analysis. So when we did this is a Manhattan plot. Okay. So you have genomic coordinates on the x-axis, p-values on the y-axis. So these peaks correspond to resistance 1. So we clearly identified a gene for the MSB1, right? And that is now assays were developed for this. This is a K-bio assay. So you don't have to work in the lab. You send DNA, they send you back the data, and you can very clearly see the 3 genotypes for that class. So that was done, and it was tested in the field as well, and almost 95% of the time we were able to rightly identify a plant which is MSB resistant. The other trait is QPM. Are you aware of QPM? Quality protein maize, which is rich in lysine and tryptophan, and that is because of the gene OPEC2 along with few other modifiers. Okay. So what we did, we had around 300 lines with us. Okay. Out of that, around 60 or 70 lines were QPM lines. Okay. So we coded the QPM lines as 1 and the rest of the lines as 0. That is the phenotype that we gave. Okay. And the genotype information was that of these 2 million data points. Here. Then we tested whether this can identify the OPEC2 gene. You can see the clear peak here. Does anyone know where OPEC2 gene is located in maize? It's on chromosome 7.01. Okay. So you see a very clear peak here, and if I check what is the underlying gene for this peak, it's OPEC2. Right, so it clearly this analysis identifies OPEC2, but what we were more interested is, other than OPEC2, what other genes may be involved in lysine. So we were looking at these small peaks also. I'm sorry, I think you are not able to see that, but then there are also peaks here, right? So we were just looking what these peaks mean. This is one of the points from chromosome 10, and then we were looking at the maize annotation fact, right? If you see this particular SNP is flanked by 2 genes. One is called Porin2, the other one is called MYB2. Yes. Then I just searched, Googled whether these genes are associated with tryptophan content, because that is my phenotype, right? A paper published in TNAU in 1998 very clearly tells you that MYB homolog is associated with tryptophan gene expression. See, to get this kind of resolution from a mapping experiment, if you use biparent populations, you need to analyze at least 10,000 plants in one population. Whereas in this case, we used only 300 lines. So that is the power of association mapping. Similarly, another high-rise point flanked by 2 genes, and one of the genes were associated with okay. Okay, so that's about a few simple traits. Okay. You can ask me what about complex traits. So as a genetics student, you should also be aware what is the current debate going on outside, within the research community, plant breeding research community. So everyone is trying to understand what is the genetics of drought tolerance, because water scarcity is becoming a major issue, right? So we are all trying to understand what is the genetic architecture of drought tolerance, or low nitrogen tolerance, or soil acidity problems. Okay, what is the genetic architecture? Can we use the same approach, a molecular marker based, QTM based approach, to improve for these traits? What do you guys think? Can we tackle drought tolerance through QTL approach. Okay, I'll get to that. So what we did initially, we did a small drought tolerance experiment using genome-wide association models. We had around 300 lines. We evaluated them at multiple locations. We genotyped it high density, and then we did the same association analysis mapping. And we found some very clear peaks. Okay, but then these peaks were not explaining very large proportion of variance. Only smaller proportion of variance, which is understandable because we know that drought is not a 1 or 2 gene trait. It's highly a complex trait. There are many genes, but it's not so complex that all 30,000 genes, there are only 30,000 genes in maize. Not that all 30,000 genes are involved in drought tolerance. So if you do a mapping experiment, I am sure you will get few regions which are more important than others. So that's what this experiment did. And we were looking at the candidate genes identified by those peaks. There are some very, very interesting candidate genes related to ethylene sensitive pathways, annexin pathways, aminotransferases. These genes have been extensively used in the private sector in the genetic engineering experiments for conferring drought tolerance. Okay. And also as a method of validation, because GWAS analysis is little crude. Okay. All associations that we find need not be true. So we need to validate those associations in the independent population. So that's what we did through 2 or 3 biparental. That's where biparentals are very important. And we found that some of the QTLs here, they overlap with your association mapping formulas. So that gives you confidence. And what is the guarantee that this method will help in hybrid development? Have you heard about this product? It's called Artesian. So this is a product from Syngenta. It's a hybrid, maize hybrid. It was released in the US for the Corn Belt, what we call the Midwestern Corn Belt of US. And this hybrid was actually developed through association mapping, and it was based on very crude association mapping. It was not even like 2 million data points. No, they just, because right now if the product is in the market, they would have started this experiment some 8-9 years back. Right? So that time all these high density genotyping things were not common. So they simply used few candidate gene based markers, but they used it so well that they ended up with a product. And it's actually doing very well in the US markets. This was developed through association. Okay, and similarly we are also getting some good leads for low nitrogen tolerance. Some of the leads, like some of the genes identified by this association analysis, there is good proof that they are involved in nitrogen tolerance. Okay, so like any approach, this association mapping also has significant disadvantages. Can anyone tell to break the monotony? Can anyone tell what are the drawbacks of association mapping? What have you read? False positives, right? Yeah, many of the associations tend to be spurious. Why? Because there is a big imbalance here. You have only 300 odd lines, but then the markers are 2 million markers, right? So by sheer coincidence, you can get some markers with high p-value, with significant p-value. So they are not really actually associated with the trait. They are kind of artifacts, what we call false positives. But then, what I always suggest people is, it's easy to validate it. If you have few good biparental populations, you can easily validate such. You can eliminate those spurious associations. For me, another important thing is, should we use single location phenotypes or multi-location phenotypes? For mapping, you use phenotypes, right? How do you generate phenotypes? By evaluating it at multiple locations. So if you use 3 location phenotypes, I get one kind of result, okay? Important peaks. Suppose for the same trait, I added 2 more locations. I made it 5 locations thinking that the data is going to be more robust, but then now I am seeing a different result. So what it means? It means there is a lot of G by E, so I am not able to identify the right region. So how many locations are actually enough to identify right gene opinions? So these are important researchable issues in GWAS. Okay. So these are all not yet well answered questions. Similarly, another important thing is rare alleles. So if I have 100 lines, one SNP has 2 alleles. So one allele is present in 95 lines, another allele is present in say 5 lines. That is 5% minor allele frequency. So with 5% minor allele frequency, it becomes difficult, though it may have a real effect on the phenotype, but then it is very difficult to identify that same allele in this GWAS p-value based association studies. That's another problem. That's why the Cornell group came up with different population structure. Does anyone know? It's called nested association map. Have you heard about it? NAM population.